it's there you go. So it's my great pleasure to invite and introduce Erin Teich for this talk. And so she has been doing a lot of very, very cool things. And so she did her PhD in the group of Sharon Glotzer at the University of Michigan, working on various problems on glass formation and packing of colloidal systems. Previously, before that, she got her bachelor's in science in physics from Brown University and also spent a year working with the AmeriCorps. Currently, she is a postdoctoral fellow with Professor Dani Bassett's group at University of Pennsylvania, and she's been looking, sort of taking some of the work that she's done previously and the tools using ideas from statistical physics and network science to investigate other problems, and I guess some of which we will hear about today in organization of also not just disordered systems, but also potentially tissues and living matter. And one thing that what I learned recently is that she's also going to be starting as a faculty in physics at Wesleyan College in Massachusetts in this fall. Another point that I wanted to say before giving the floor to Erin was that she has been a very, very vocal advocate for increasing equitable or increasing platforms for equitable science, and particularly using data-driven tools and techniques to analyze and to perhaps even address disparities in gender and racial disparities in science, particularly with regard to academic citation. So some very, very cool set of ideas and thoughts and scientific work. And so with that, I'm very excited to hear what she has to tell us about crystallinity in white matter. So take it away, Erin. Thank you.
Great. Thank you so much, Suraj. That was a really kind introduction. I really appreciate it. So let me make sure. Okay. So yeah, I'm really happy to be able to talk to all of you guys today. I'm very honored to be able to participate in this seminar and discuss a little bit about a recent paper, sort of a recent work that I've done as a postdoc at Penn on soft matter in a sort of a slightly different context, maybe than perhaps this seminar typically deals in, which is maybe a more sort of neuroscientific context. And particularly, what I'm hoping to communicate with this talk today is that tools from soft matter physics and from material science might actually be extremely useful in a sort of neuroscientific regime, and especially for, or specifically for characterizing white matter in the human brain. So I'm going to discuss some examples of ways that I think soft matter is helpful in that context with this talk. So the main thesis of what I'm gonna talk about today, I sort of wanted to sum up with these two juxtaposing images from the worlds of soft matter and material science on the left here, and neuroscience on the right. And I want to again, sort of emphasize that the overarching sort of theme of what I'm going to speak about today is sort of forming a bridge between these two worlds. So on the left is this really beautiful image of a self-assembled clathrate phase from gold triangular sort of bipyramidal nanoparticles. And on the right is sort of a beautiful anatomical engraving of a brain by a British physiologist centuries ago at this point. And so what I want to sort of convince everyone here of is that tools that we sort of know and love from the world of material science, used to characterize phases like this image on the left, can actually be used to characterize tissue architectures in this image on the right. And thus are sort of worth porting over from the world of soft matter into the world of neuroscience. So first I want to emphasize that at the heart of both fields, material science and neuroscience, there's this tight coupling between structure and function, meaning that structural characterization in both fields is completely necessary in order to understand and design function in the case of material science. So in the world of materials, I think we're all very well aware of this tight coupling and its importance. And so I'll just show a few motivating examples. So this figure is from a really beautiful review article that was written not too long ago. And this particular image is showing this sort of array of binary nanocrystal superlattice structures shown in these photos here. And below here is showing how various material properties like photoluminescence, magnetism, and even electronic and catalytic properties can sort of change when we change the structure of these binary superlattices, right? So this structure function coupling on this length scale is quite real and quite useful for designing functionality. This tight coupling doesn't only exist in human-made materials, right? We're all well aware that it exists as well in biological materials, including, as an example, viruses, right? So this is we're all unfortunately very aware of that. This is a really beautiful illustration of the Ebola virus. And the functioning of this virus is completely supported, right, by the structure of this virus, right? And on a simpler scale, lipid bilayers, for example, can have a variety of morphologies. And these different morphologies basically underpin cellular functioning, right? So this tight structure function coupling is happening in materials all around us in a variety of contexts. The same is true within neuroscience. So the brain is a biological material, right? And specifically, I'm going to be discussing the human brain in this talk. And neural architecture completely constrains and in many ways actually determines brain function. So first I thought I'd provide a super brief primer on brain structure, just to get us, and a very overly simplistic primer at the level of my understanding of brain structure, just to get us on the same page. So it can roughly, very roughly, be divided up into two categories: brain tissue, gray matter and white matter. Gray matter consists mostly of neuronal cell bodies. So this parts of the neuron here, mostly distributed over the surface cortex, so on the outside of the brain. And this matter is responsible for a lot of how our brain functions, including muscle control, our perception, our memories, our emotions, how we make decisions, and so on. And then white matter is the tissue beneath gray matter that's composed of axons or axonal tracts. So these sort of parts of the neuron here that transmit chemical and electrical signals between the neuronal cell bodies that make up gray matter. So these axons are literally the message passing highways upon which a lot of our brain function relies. And I just want to emphasize that we have a staggering amount of these highways by the time we're 20. So we have around something like 150,000 kilometers of white matter axons by the time we're 20. So this is a staggering infrastructure, right, that supports the way that we function. And maybe one other thing I'll say about sort of in this super broad overview of brain structure is that the development of white matter is pretty decoupled from the development of gray matter, especially as we age. So if we just focus on these three images here, the actual volume of our gray matter declines slightly on average from the time we're 5 to the time we're 30. But the average volume of white matter actually grows and sort of saturates by the time we're 30. So in a very broad sense, this is indicating that our brain is becoming increasingly connected to itself as we age, and this connectivity is supporting the increasingly sort of complex ways we can function as we age during this period. So the structure of both gray and white matter determines in some sense function. So in the cerebral cortex, so with gray matter, areas that are segregated according to cytoarchitecture or cell structure tends to correlate with function. So this image is showing these segregated areas called Brodmann areas and their correlated functions. So as an example, right, the primary visual cortex is a Brodmann area that sort of tends to have the same cellular structure, the same with the primary motor cortex, for example, or the primary auditory cortex, and so on. And so all of these areas sort of have some kind of correlated function, right? I should emphasize that this map was formed or was first proposed in 1909, was first discovered in 1909. And so the picture has considerably become more complicated since then, right? There's not a strict one-to-one correlation between structure and function in the cortex, but there is nevertheless sort of a correlation generally.
Can I ask a question?
Please.
Maybe motivated by what you just mentioned, given the not so quite clear one-to-one map between structure and function. What happens when you have damage to brain tissue, and given that brain plasticity lets different regions of the organ to take over function? Are structural changes also seen, particularly to precede functional changes? And or is there a correlation? In the cortex, yeah?
That's a great question. I don't know. That would be a great question for a neuroscientist.
Okay.
Yeah, I really don't know. That's really fascinating. I can't even speculate. I mean, so what I'm going to discuss, sort of for the rest of the work is white matter. So, different from this cortical matter. And actually I'm about to discuss how damage to white matter, does obviously correlate with loss of function, and there's some plasticity there in terms of rebuilding those connective pathways. But yeah, in the cortex, I really don't know. Yeah.
Okay. No. Thank you.
Yeah.
And with the white matter, and I mean, this might be very, very basic things which I'm missing. So you had these plots of increase in volume as a function of age, right? And so the white matter keeps growing as you grow older. Is that correct? But there was also a different row at the top. So maybe what was the row at the top versus the bottom?
Yeah, I should have mentioned. Is just see how the scale is different. So, and these are both in years. So this is illustrating that when we're in infancy, our brains are just growing. And so the volume of each of these components is correlated. But then when we sort of enter adolescence and adulthood, this becomes decorrelated.
Okay. Thank you.
Yeah, thank you. Yeah, so those questions were a nice bridge to what I'm sort of mentioning in this panel, right? Which is that beneath the cortex, there's also a structure-function coupling. So structural damage of white matter pathways due to disease and aging correlates with loss of function. So diseases like Alzheimer's or multiple sclerosis, which I'm showing some images of damage from these two diseases here, and schizophrenia are all sort of marked by white matter microstructural damage and consequent disrupted communication. between regions of the cortex. And so this is all sort of a motivation for why reliable and thorough structural characterization of white matter is completely necessary in order to fully understand actual brain function, as we develop, as we age, and perhaps as we are impacted by these types of diseases. So structurally characterizing white matter is going to be the subject of the rest of the talk. So now let's sort of provide a brief outline of the talk. So as I mentioned, I want to sort of demonstrate that material science provides a unique set of tools for characterizing white matter. So first I want to discuss how white matter is actually reconstructed non-invasively in living human brains, using this beautiful imaging technique called diffusion magnetic resonance imaging. And then I want to discuss 3 tools from material science that we used in this paper to analyze white matter in the context of these diffusion MRI scans. So first, we measured crystallinity or structural homogeneity of white matter throughout sort of these brain scans, and found that this is an anatomically relevant, reliable, and novel sort of measure of white matter microstructure. We then sort of did a user-related tool of segmenting white matter microstructure into crystal grains or sort of parcellations of similar structure. And we found that this had sort of considerable overlap with other white matter parcellation schemes that were developed by totally different means by neuroscientists and other papers. And finally, if I have time, I want to show that characterizing the shapes of local white matter structural signatures using bond-orientational order parameters, again, kind of taken from soft matter physics, is also a very useful means through which to characterize structure. And it can be used to detect crossing white matter fibers and especially streamlined regions of white matter and so on. So first, I want to just discuss how white matter is reconstructed. It turns out that water diffusion is actually a proxy signature for white matter tracts sort of throughout the brain. And this is because white matter axons, so that these parts of the neuronal cell or axon sort of bundles of axons, inhibit the diffusion of water more severely in directions that are orthogonal to the way these axons are oriented. And they inhibit the diffusion of water less severely in directions that are parallel to the way these white matter sort of axonal tracts are oriented. So both inside the axon itself and outside of it, effectively, sort of the myelin sheath of the axons, their cell membrane and biological architectures like microtubules and neurofilaments inside the axons, all of which are oriented parallel to the way that white matter is oriented in a specific region of the brain. They act as obstacles for the diffusion of water in sort of these perpendicular directions. And they don't obstruct the diffusion of water in these parallel directions. So I want to sort of emphasize that, as with all, I think, biology and neuroscience, the story is a lot more complicated than what I, as a physicist, might have wanted. So, it turns out that both intra-axonal water diffusion, so inside the axons, and extra-axonal water diffusion contribute to this diffusion signature that acts as a proxy for white matter. And it's not clear which one is sort of more deterministic or which one contributes more to this diffusion signature. The way these axons are sort of bundled, how tightly they're packed, how much that package is sort of splayed, as indicated here, also really matters. And generally, sort of these are electron micrographs of cross sections of axonal bundles. it's not just sort of these nice sort of, axons aren't just these nice tubes that are channeling water diffusion. It's a lot messier than that. But the takeaway message is that water diffusion is anisotropic around these axons, and that anisotropy, if it can be measured, indicates white matter orientation. And so diffusion magnetic resonance imaging is a way to measure that anisotropy of water diffusion. So it's a way to non-invasively characterize white matter microstructure as a result. So this is an imaging technique that tracks proton spins within water molecules over a given time interval at quite a high spatial resolution of around a few millimeters. And what neuroimagers do is they effectively carve up the brain into cubic regions, or I'll use the word voxel from now on, that are a few millimeters across. And they build probability distribution functions within each voxel, indicating the probability that a water molecule at the center of the voxel has diffused in a particular direction, at a particular orientation over a given time interval. And so these sort of probability distribution functions are thus giving some indication of white matter structure in that particular region of the brain. And different sort of white matter microstructures are going to give rise to different diffusion probability distribution functions, or PDFs. So if the white matter is oriented especially randomly inside this cubic millimeter scale region of space, the PDF is going to be fairly isotropic. If instead the white matter is oriented in a streamlined manner, in an especially unidirectional manner, where there's sort of this bundle of axonal tracks that are all pointing in the same direction, that's going to be reflected in the PDF. And the same if there's sort of a crossing.
Okay, welcome everyone.
Bundles of axons. That'll also be reflected in the PDF. In practice, this is a lot of data. This is like a 3D probability distribution function in tens of thousands of voxels that make up one scan of a human brain. And so typically people reduce the dimensionality of that information. And what I'm going to focus on especially are these ODF signatures of these probability distribution functions, or orientation distribution functions. So these are built from PDFs by just integrating out essentially over R, but retaining all of the angular information of these distribution functions. And so the signatures at the end of the day look like, well, their surfaces, you can think of them as deformed spheres, where the distance from the center to each point on this deformed sphere is indicating the probability that water has diffused in that particular direction. So these are sort of directions of high diffusion probability.
Erin, can I ask another question over here?
Yeah, of course.
So this ODF isosurface is for each voxel, right?
Mm-hmm. Yes.
And how is this time window chosen for constructing the diffusion probability density?
That's a great question. I mean, I think there's a lot of considerations that go into it. I think, so this is a technique that's been in development, I think, since the 90s, probably. Okay. It's used clinically, but the strength of the magnet determines what spatial resolution you can give. So I think probably the time interval is chosen in concert with the strength of the magnet and some knowledge of probably the diffusion constant of water. Okay. You know, in order to just try to probe like the highest spatial resolution possible. But there are like a lot of different sort of imaging paradigms that people use. It's almost like a scattering experiment, but you're scattering off of water or something, you know, it gets quite complicated. And the other thing I want to say is that it's also interestingly just totally constrained by human limitations. So you would ideally want to have someone in the scanner for a really long time, but especially for kids, for example, it's like, you know, they're not going to hold still for that long. So anyway, everything's sort of a delicate balance.
Yeah, of course. Yes. Okay. Thank you though.
Yeah, of course. Yeah, it's a really fascinating technique because it's so, I feel like the physics is so beautiful, but then it's so messy because it's you're literally trying to scatter off of someone's real like brain. Yeah. But yeah, so at the end of the day, you know, if neuroimagers have done their job correctly, and you know, very usually they have, a brain scan will basically consist of a set of ODFs, one per voxel, one per small region of volume in a brain. And these are simplified structural environments characterizing water diffusion in each particular region. So I'm showing here what an array of these ODFs might look like in practice for one diffusion scan. So these are two sort of views of the same scan, and these are sort of close-ups of a particular, you know, region of that scan. And so you can see each of these little signatures is an ODF that characterizes diffusion in that particular region of space. And so maybe by eye, you can already see that these seem to really correlate with probably the way that white matter is oriented in the brain. You know, you can see already just from sort of linking the ODFs up together by hand that there are these major white matter tracts moving throughout the brain. And so this one, for example, that I'll talk about a lot is the corpus callosum, which is basically a saddle-shaped set of white matter tracts that connects the two halves of our brain together. So here I'm just showing a different sort of slice of these ODF signatures for a particular example brain scan, and a reconstructed ODF on the left here. And in practice, what we actually do is we extract the sort of peak vectors of each ODF and actually use that set of peak vectors. So those are these red lines here, as structural signatures of white matter in each voxel. So we reduce dimensionality even further, and we just consider these sets of vectors. In the interest of time, I'm going to skip over this slide. This is just introducing the data set that we actually used as sort of our sandbox for playing around with how metrics from soft matter and materials science can be used to analyze white matter microstructure. But what maybe I'll emphasize is that this is a data set that was collected several years ago, actually. And we were just very lucky enough to be given access to an anonymized version of this data set. It was originally used to measure the relationship between cognitive resilience and sleep. And so a lot of different measurements were taken of human subjects in this data set. But luckily for us, one of those measurements was sort of these diffusion MRI scans. And they were taken for 30 subjects, 8 scans of each subject taken every 2 weeks. So this is a really beautiful data set because it's gonna show white matter microstructure both across different subjects and across different scans of the same subject. But we're not interested in sleep resiliency. We just are sort of using this as a playground. So first, let's discuss actually analyzing structural homogeneity of these ODF white matter signatures. In usual particulate materials, people often define local environments or neighborhoods, right, of particles, and they use those neighborhoods to say something about the local structure and materials. So one way you could think about defining a local environment is just by considering a set of vectors pointing from each particle to its nearest neighbors, right? That's an especially simple way of defining a local environment around a particle. And what one could then do is compare these local environments, sort of the local environments of neighboring particles to each other. And if those local environments are similar enough, you might say, okay, these particles occupy the same small crystal grain, maybe in this material. They're especially crystalline if these neighborhoods are similar enough. More specifically, one can sort of compute a mean squared difference, right, between these vector sets, and use that difference as a proxy for how similar they are, right? So if this difference is especially small, if this delta parameter is especially small, then these vector sets are especially similar. So this quantity inside these averaging brackets is just the magnitude of the vector difference between particle i's mth environment vector and the matching paired vector from particle j's environment that basically best matches to RIM. And this average then would be taken over basically the best matching, over the set of best matching pairs between all of these environments. And so then from that measure of dissimilarity between two neighboring environments, one could construct a per particle average dissimilarity that every particle environment might have with respect to its neighbors. And we choose in this paper to non-dimensionalize it, to normalize it in sort of this manner. So here now, this average is taken over all of particle i's neighbors j. So this is the average dissimilarity that particle I has with respect to all its neighbors. And this it's being normalized by the average size of particle i's environment. So this notation's horrible, but these average brackets now are over all of the m environment vectors in particle i's neighborhood, right? So when this normalized quantity is high, then this particle has especially high dissimilarity to all of its neighbors. And when it's low, this particle I is especially similar or to all of its neighbors, or especially structurally homogeneous or crystalline. And so we can port over this quantity and this technique exactly when characterizing the spatial homogeneity of these ODF signals that indicate white matter structure. So for neighboring ODFs now, we extract sets of peak vectors for those ODFs, Ri and Rj, and we compute this dissimilarity between them in exactly the same fashion. We just port over this technique exactly. And so what we find, or effectively what we're computing, is a measure that's indicative of how spatially homogeneous these ODF signatures are with respect to their neighbors, right? So how spatially homogeneous white matter microstructure literally is in this small local neighborhood of the brain. And so I'm showing an example map of this crystallinity metric, or this delta metric here. So when this dissimilarity is especially low for these more white values, crystallinity is especially high. And so one can see now, so each voxel now is just a square, right, colored according to this value. And there are some really nice sort of white matter mesostructures that are sort of popping out of this scan. Again, we can see our friend, the corpus callosum, is sort of registering as an especially crystalline region within this slice. I think the usefulness of this metric becomes even clearer when we look at the structural homogeneity of the whole brain. So this is for one subject in the crash data set. And we can see now that there are large white matter tracts that register as especially crystalline, distributed throughout the brain. The corpus callosum again is sort of registering as a especially crystalline region. Voxels now are spheres in these particular images. These are 3 images of the same scan. And the color is according to this delta parameter averaged across all scans of one subject. And so, yeah, so we can see both sort of highly crystalline regions and almost like grain boundary regions around these especially crystalline regions of the brain. So this is just a histogram of those values, and it's sort of illustrating our color scheme. We can compare our crystallinity metric delta to other common metrics that characterize white matter via diffusion MRI techniques. So these plots are just showing 2 comparisons between our metric and 2 common diffusion metrics, MD, which is mean diffusivity, and GFA, which is generalized fractional anisotropy. So these are joint histograms that are accumulated over all voxels in one scan of this particular subject. So mean diffusivity is a scalar that indicates the just basically the overall isotropic magnitude of diffusion within a voxel. And generalized fractional anisotropy is indicating how anisotropic that particular signal is within a voxel. And it's just basically like the variance of the ODF over its surface. So high GFA here is indicating something sort of a diffusion signature that's very anisotropic. So we can see just in this one particular joint probability or joint histogram that there are some trends in terms of how these other metrics are related to crystallinity, but the relationship here is far from one-to-one. And we can quantify these types of relationships much more precisely by basically just calculating a Pearson correlation coefficient between each set of distributions for each scan in this entire data set that we have. And then just showing this distribution of correlation coefficients over all scans for the relationship between crystallinity and mean diffusivity, and crystallinity and this other, anisotropy measure. So what we see is there's this sort of weak correlation, positive correlation between delta and MD, and there's a stronger negative correlation between delta and anisotropy. So this negative correlation is kind of interesting. It implies that voxels that have high anisotropy tend to exist within sort of streamlined white matter regions where water diffuses in the same direction through neighboring voxels. And thus these voxels have high crystallinity, right? High sort of homogeneous structure and low values of delta. But in contrast, as we can even see in this particular joint histogram, voxels that have lower values of anisotropy tend to have very wide ranges of crystallinity, showing that essentially this delta parameter, crystallinity, provides information that's pretty distinct from diffusion anisotropy, especially in areas that are not isotropic, right? That don't contain streamlined white matter.
So there's a question on the chat, if I may.
Oh.
Yeah, so from Miguel, who's asking, just out of curiosity, would 16 weeks be enough time to try to see any evolution in the microstructure of the white matter?
Yeah, that's a good question, Miguel. So I would say no, especially because these are all scans taken from adults. So I didn't mention it, but all the scans were from basically subjects who are in their 20s. And so when people are around that age, I don't think 16 weeks is enough to see see any evolution. And in fact, we were happy for that, because we could actually use then those multiple scans of the same individual to test how reliable our metric was across basically like multiple measurements of the same individual. So we weren't expecting that the microstructure would be changing at all over time. We were basically using them as, just measurements and remeasurements of the exact same structure. Another question is, crystallinity always highly asymmetric about the middle brain, right and left half of the brain? So if I'm understanding the question correctly, I believe that it's usually symmetric. Oh, about the middle plane. Yeah, I believe it's usually symmetric about the middle plane. So I think that can be shown here if I'm interpreting the question correctly. Essentially, if you look at this slice, for example, these two halves seem pretty symmetric, right, with respect to that plane. Okay. So I think in the sort of interest of getting to some of the other metrics that I mentioned, I won't spend a lot of time on this at all. Basically, using this data set, we were able to compute some measures of test-retest reliability for our measurement of crystallinity versus other common metrics. And we were also able to compute some measures of across subject variability. And we found that our metric of crystallinity is pretty reliable across scans, which is what I was mentioning to Miguel. That's why we could use those multiple scans of the same individual. But it seems to be quite variable across individuals. And so this is actually a really nice combination of features, because it means that crystallinity could possibly be useful to differentiate white matter in some kind of broader data set that might feature actually, changes in white matter along different ages, or as we age, and as a function of, different diseases that we might have.
I think there's a question from Tim.
Yes.
You are still muted.
Thank you so much. It really all interesting. And I don't know how much more of your talk or how much time we're going to have. So I just wanted to ask, when would be a good time? You're generating so many, I'm kind of naive to this technique, and I kind of had a list, and just wondering if there's some place I could, some way at the talk here to just kind of go through those, and you pick out what would be important for the work that you're doing. Can I say something here, or would you like me to wait?
Oh, you're welcome to say something here. Yeah, of course.
All right, so can I just read through it? And please, this is not my area, but it's so interesting. Can you distinguish bound water to particle diffusion from free water diffusion in this technique?
Do you mean generally using diffusion MRI?
Well, this here, you're talking about the anisotropy and the crystallinity and some of these characteristics, but wouldn't bound water and free water have different signatures? And if a particle is moving across in the axon, for instance, there would be water bound to it as well as free water. That would be highly dependent on salt solutions and stuff like that. So can you see those differences, for instance?
Yeah, I'm not sure. I would love to talk more with you about that, just to make sure I fully understand the question, because I think probably you know a lot more about neuroanatomy than I do.
I'm a physician, so, that's why. Yeah, but
yeah.
One other thing, and I would like to sometime maybe, because I got like 8 questions you've generated. The other one is, can you distinguish, I'm a little confused how you distinguish amorphous from crystalline areas for MRI, because I thought the MRI was particle to particle interactions. And those generally are affected, as I remember, I hope, by the general surrounding milieu of it affecting the residents, as opposed to a distinct either directional or repeating structure. And so you mentioned about crystallinity. And just one other thing, and can you just, how much of this is structural in the brain, right? Supporting girders and rather than functional tracks. So the looking at the metabolism of these structural areas, which don't really relate to function as opposed to function. So I do have these things. I just throw those few out there, and maybe at some point I could contact you, or you could, if you have.
Yeah, absolutely. Yeah, I would love to talk more offline. Yeah, I guess I'll just say, certainly we're only looking at structure in this data set, right? Like there's nothing about functional MRI that we're looking at, only looking at water diffusion and saying that's a proxy for, white matter orientation. And then again, yeah, this measure of crystallinity is really just looking at the homogeneity of those white matter signatures.
Yeah. I would just throw out that I would expect those transitions at some point to be temperature dependent. And so that might indicate more functional aspects. But you say you're looking at the structure, but some of the structures may affect the function. So if they're permanent, then all right. Thanks for taking my question.
Yeah, no, of course. Yeah, thank you.
If you put in the chat a way to contact, I'd appreciate it. Then I can.
Of course, yeah. I have my email address at the end of the talk, but I can also.
All right, thanks again.
We'll also have time for discussion for 15 minutes after the talk. So I suggest we go ahead with the talk. We have like.
Oh, okay. Yeah, that sounds good. Okay. Yeah, so this is just perhaps sort of a final deeper dive into how crystallinity varies throughout the brain. So here I'm showing a really beautiful map, I think, basically encapsulating all of the scans in our particular data set. So this is crystallinity now averaged over all scans and all subjects. And we can see that it varies smoothly throughout the brain. And here I'm just showing a thresholded version of this map above, showing just sort of the top 10% of crystalline voxels in the brain. And these more crystalline regions are localized towards the center of the brain, and they contain major streamlines of white matter, including this giant saddle-shaped corpus callosum. So we can do a little bit of a deeper dive even further and now use a white matter atlas in order to consider sort of segments of voxels that are actual sort of anatomical regions according to this commonly used white matter atlas, and then pool essentially crystallinity values calculated basically in every region over all subjects and all scans of this data set, and sort of tease out anatomical regions of the brain that are especially crystalline, containing low values of this parameter here, and colored in white here, and that are especially dissimilar, containing higher values of this parameter. So the story really is the same, right? Like these more crystalline regions are located towards the center of the brain, close to our sort of connective pathways between the two hemispheres, and the least crystalline regions seem to be located maybe more towards the back of the brain. Okay, so now I will very briefly turn to a related tool that we also thought was pretty interesting in order to characterize white matter microstructure. And this is crystal grain segmentation of these regions of white matter according to similar structure. So first I thought I would demonstrate the method by which we use to calculate these grains. but maybe I'll be fairly brief about it. Essentially, to parcellate our scans into crystal grains, we formed a weighted lattice graph for all of our scans, where voxels were now nodes in this graph, and there were edge weights between neighboring voxels or nodes, and the edge weights were related to how similar the ODF signatures in neighboring voxels were to each other. So higher edge weight meant there's more structural similarity between neighboring ODFs, and lower edge weight meant that there's less structural similarity between neighboring ODFs. Here's sort of the expression for our edge weight, but you can just sort of essentially consider it as sort of the inverse of this dissimilarity, right, between ODFs of neighboring voxels. So when this dissimilarity is very high, this edge weight essentially goes to 0. And when this dissimilarity is very low, and the neighboring ODFs are about identical, then this edge weight goes to 1 and it reaches a maximum. We could then sort of do some clustering of voxels in our weighted graph in order to essentially identify communities of nodes that are densely intra-connected, meaning that they have especially similar structure to each other, and sparsely interconnected, meaning that they're pretty dissimilar from neighboring communities. And so I'd be happy to go into detail regarding this actual algorithm if people are curious. But the end result, right, is this sort of parcellation of voxels in each brain into spatially contiguous sets that have very similar structure among them. So now I'm showing voxels here colored according to whether they're in the same community or not. I'm only showing communities that have a population of 10 or greater. And we can see now, again, that there's sort of these mesostructural features, these white matter sort of features that pop out of this example slice of ODF signatures. I think this example image gives a better idea of how crystal grains that are computed using this method actually look in a full brain scan. So now all crystal grains in this particular image are colored the same if they have a population greater than 100. And we can see that they're sort of roughly symmetric across hemispheres, roughly. And they are sort of branched, right? So they follow this sort of anatomical branching of large white matter tracts. We can sort of use this crystal grain parcellation in, I think, interesting and informative ways. Here I'm showing some of the crystal grains sort of towards the center of the brain and kind of an exploded view. And we can see this rough hemispherical symmetry in a little bit more detail. We can also see sort of composite environments in each grain. So these are basically ODF signatures for all voxels in each crystal grain, superimposed on top of each other and represented as sort of ball and stick. So you can get a sense of the similar white matter microstructure that's in each of these grains. We can get a handle on whether or not our crystal grain segmentation is useful as a parcellation scheme by comparing it with a commonly used white matter parcellation that was generated by hand many years ago in previous work, very unrelated to our segmentation scheme. So basically, we first create this weighted lattice graph for each subject in the crash data set by averaging edge weights over all scans of the same subject. And then we can find crystal grain segmentations for each subject over a variety of resolution parameters, gamma. I sort of skipped that in the a previous slide, but it's just sort of giving an indication of how big or how small we want our crystal grains to be. And we can measure then overlap between our crystal grain segmentation and this common white matter atlas using different metrics for partition overlap. So I'm showing two here. This is adjusted mutual information between our segmentation and this white matter atlas, and the adjusted RAND index between our segmentation and this white matter atlas. And so we see that it sort of indicates partial overlap, right? And it seems to peak at this range of resolution parameters. And if we investigate this sort of by eye, and we look at our crystal grain segmentation that has the highest overlap with the white matter atlas, we can see, so this is the white matter atlas here. This is our crystal grain segmentation here. We can see, again, sort of just to eye, that we seem to be grabbing crystal grains or sort of parcels of white matter that match somewhat with this white matter atlas. And we can make this comparison even more explicit, basically by finding the best matched crystal grain to every white matter atlas region, and the matching maximizes the total number of overlapping voxels between all matched pairs. So that's just a sort of a quick and dirty way of visually comparing our segmentation, even more completely to the atlas. So now I'm sort of coloring the segments such that they match best with the white matter atlas, and hopefully you can see even more clearly that we are definitely getting some grains that match particularly well with the atlas. And so so here I'm showing our crystal grains that have especially high overlap with regions of this white matter atlas. It's pretty nice. Some of these regions really match especially well. And for transparency, I'm also showing some of our crystal grains that have especially low overlap with this white matter atlas. But so the takeaway, I think, is that since we have partial but not complete overlap with this white matter atlas that was formed in a totally different way, we think that crystal grain segmentation is might be a really nice data-driven microstructural feature that could be used in concert with some of these other white matter parcellation schemes that neuroscientists and neuroimagers have used in the past. So maybe I know we've hit an hour, so I can sort of skip over this final section. It was sort of the smaller one anyway. But the takeaway here is that we can also use so-called Steinhardt order parameters or bond-orientational order parameters that are often used in soft matter physics to characterize symmetries of local particle environments. We can port that tool also over into a diffusion MRI context and use it to characterize the shapes of these ODF signatures in a way that I think is pretty nice and can sort of distinguish between ODF signatures that are especially streamlined versus ones that are sort of represent fiber crossings. Versus ODF signatures that might be pretty noisy. And in a whole brain context, these parameters sort of again are anatomically relevant seemingly, and are fairly independent from other white matter metrics that are used as well, like mean diffusivity and generalized fractional anisotropy. So with that, I can conclude. I can also address some more of the questions in the chat, but hopefully I've given a super sort of maybe big picture overview of the fact that I do think that tools from soft matter physics do have potential use in the characterization of white matter microstructure. And I think we're sort of only limited by our creativity in terms of porting these tools over into a neuroscientific context. And we can use these in the future to investigate changes in white matter structure as a result of development or aging or, various pathologies or even gene expression and so on. And so if people are curious, this was published last year in New Journal of Physics. So please feel free to either get in touch with me or look that up. So yeah, so thank you for listening. And yeah, I can jump right into answering these questions if you guys want.
Thank you, Erin. It was fantastic. So clap from me and from everyone else in the virtual audience. Yes, I guess there are two questions in the chat. Maybe Miguel, if you want, you can unmute yourself and ask your question.
Okay, maybe it can be easier. Yeah, thank you very much. It was a very nice talk, Erin. So I was curious, I think you mentioned this at the end after I asked the question, but just to make sure, when I think it was slide 18 or something like that. So you were showing superimposed all these, yeah, here, right? Like, so for each of the regions, you show this superimposed diagram, right? And some of them look very similar to each other, but rotated somehow. So I would say kind of the same type of structure.
Right.
Some of them are different, right? So I was wondering if even, I mean, you show very clearly that you can distinguish different regions. I wonder if you can say even more, saying like, oh, these regions are more similar to each other, belong like to the same group or type or whatever, whereas, you know, other regions also are from a different group or something.
No, I think that's a super great question. Yeah, yeah, so it has to do, I think what you're asking is if basically when we were sort of comparing these signatures to each other, we allowed for them to rotate in order to find the best overlap, right? Whereas instead of just thinking about, you know, the mean squared difference between these environments when they're not rotated, right? If we allowed some rotation and said, okay, what's the best? Are they similar if one can be rotated onto each other?
I guess I would be, yeah, I think I understand how you do it, like to distinguish from region to region, but I would say something like, I don't know, like the two from the top of the diagram, right? Like they have like a, yeah, I don't know, like this, yeah, that one and the blue one, right?
This one
they
and
look,
this one?
yeah, these two,
Okay.
for example, right? Like the diagram that you have next to them, they look pretty similar, right? Like just,
Sure.
all of them also look similar with two of these lobes or whatever you want to call them. I don't know. But for example, the one on the top left looks different, right? It looks like photo or something like that. So I was wondering if there is any, I mean, there is a difference in the structure for sure, because you are showing it there. Could you say, oh, look, all the ones that have like two lobes have a similar function or something like that, whereas the ones that have, because I guess like the four lobes, you said it relates to more crossing between the axons or something like that,
Exactly.
right? So I was wondering if that means they have a different function or something.
Yeah, that's an interesting question. I mean, yeah, I guess it depends what you mean by function. Certainly there's sort of regions where signals cross. And so, right. And so in that, or, you know, I'm being super naive in my understanding of like brain communication, but that could be a region in which signals are sort of crossing, right? And so perhaps damage to that region is especially harmful.
I mean, yeah, I mean, that was my question. Like,
yeah,
I think it's a.
No, I think you raise a really good point. Yeah.
Okay, no, thank you very much. It was really nice. Thank you.
Thanks, Miguel.
So I guess we're going to stop the recording now, and we will continue taking questions. Andrei Zakharov, do you want to ask your question